Hello learners, welcome once again to our ongoing series on Mendelism and beyond. In my previous lecture, I had given you a summary of how the environmentally induced sex determination was determining whether it was a male or a female or an intersex or a hermaphrodite gametophyte depending upon the environmental conditions in bryophytes and in pteridophytes like equisetum. To continue the other examples, another very important example is of Ceratopteris. This Ceratopteris richardi has been very extensively worked, and it's a good example of how to link the environmental sex determination to the breeding systems. Well, the female gametophyte in this case wants to promote outcrossing. Every plant would promote outcrossing because it would mean more of genetic variation. And the gametophytes, the female gametophytes, they release a chemical pheromone known as antheridiogen. Now, this antheridiogen controls the sex of the nearby developing gametophytes. And this antheridiogen, as you can see on your screens in the fern, is going to induce maleness in the gametophytes, and thereby you will have another sex ready for fertilization. Now, this is of immense adaptive value. You will agree, learners, that if there are males also around, it would be a chance, an increase in probability of outcrossing, because the males, they would provide sperm for the females rather than the females becoming hermaphroditic themselves or bisexual, because if the females become hermaphrodite, they can at the most do self fertilization, which is again a sort of a inbreeding, and it's not outcrossing. But the advantage is that by chance, if no fertilization occurs, the female gametophyte can still become hermaphrodite and self fertilize. So that means if the conditions are conducive to growth, why should it outcross? Because then it does not need any variability. So if there are conditions which are good throughout, you must understand that it would promote inbreeding, and in the long range it would ultimately result in what we call in genetical terms as inbreeding depression. On your screens you would find a summary of how the environmental sex determination in ferns can be modulated by nutrient availability, light, competition between the plants, and of course the pheromone antheridiogen. So there is a transition, and more nutrients would mean femaleness. Less nutrients would mean maleness. So we are going from left to right as far as the transition is concerned. Another example on your screens you would find is in the form of the fern, which is called as chain fern, Woodwardia radicans. In this case, we find that if there is a nutrient supply and crowding, which again amounts to a stress, it is going to affect sexual reproduction in such a way that it would give rise to male plants, and the females only when the conditions are good. So that means harsh growth conditions always modulate the plants to turn into males, whereas the good conditions, the favorable ones, they are, that is large size, and subsequently we have the formation of the female ones, and ultimately they will even give rise to the bisexual ones. So that means the ESD, or environmentally induced sex determination, has been mainly studied, as I said before, in angiosperms. There are few examples in the lower plants which we have just discussed. But then we find that among the angiosperms, this labile or flexible sex appears to be more common among the dioecious plants and also among the monoecious plants rather than among the hermaphrodites. Let us take a few examples of the angiosperms, because you will also agree on one point that majority of the plants which are going to show flexibility need to be first perennials, because there is a flexibility of sex, and it is more important for species with long life cycles, because the ones which are ephemerals would not have no time or no chance or a time period to transform their sexes. So hermaphrodism is the most common and probably the original mode of reproduction in seed plants. Learners, you should try to appreciate the evolution of the sex determination mechanisms, how they appeared in the past, and whether what is their lineage or their relationship with the modern time or the modern extinct species of lower plants as well as the angiosperms. Because we know that the species with unisexual flowers have evolved repeatedly from hermaphroditic progenitors, and therefore the mechanisms which are controlling sex expression in seed plants naturally also tend to be very diverse. It has been estimated that only 4% of the angiosperms are dioecious and the 7% are monoecious. So that means there must be some advantage of being dioecious or dioeci as we call it. So dioeci is infrequent, right? Only 4%, but then it is widespread among the angiosperms. And in gymnosperms, we would find that dioeci is a more frequent condition than in angiosperms. And all species, of all these species, approximately 25% are the dioecious ones. And the reversal, as we find the reversal of sex by using certain chemicals, certain hormones in higher plants, it also indicates that there is a lability or flexibility in the interaction between sex determining genes and the physiology of such species. And we know, although, that in many dioecious species sex is rigidly controlled by genetics, although only a few species they possess sex chromosomes, not all of them. And we also must appreciate that sex chromosomes have evolved so recently that many dioecious species with sex chromosomes have wild relatives which are hermaphrodites and without sex chromosomes in the same genus. So that means in the same genus we have representatives which are with sex chromosomes and ones which do not possess them. On your screens you would find an example of the male plants of hemp, that is Cannabis sativa. Now this differentiates into a female plant or an intersex by the treatment of ethyl hydrogen 1 propyl phosphonate, and one can modulate the type of sex of a plant as it would assume in future. There are also examples in the flowering plant Catasetum, where again you can modulate the sex of the flowers by the hormones. Environmental sex determination is also with reference to size. As you can see on your screens, the beautiful plant of Jack in the pulpit, which is giving the appearance of a snake, which is Erysima. on your screens you can find that the small plants, they will be male, whereas the large plants would be females. So that means here the size would naturally be depending upon the kind of conditions in soil and the light and of course the cluster effect. In cucumbers also, in melons also, we find that the sex of the plants can be modulated by temperature, day length and various hormones. In addition, we find that there are various ions also which are modulating the sex of an individual. And these ions are, say, calcium divalent cations and the magnesium divalent cations. Just to summarize, the cytokinins are going to trigger the male flowers here, and ethylene and GA, that is gibberellic acid, they are going to promote the production of female flowers in such angiosperms. We find that the terminology of describing the angiosperms as far as their sexes is concerned is somewhat confusing and requires a review, because we know majority of the flowering plants are perfect. Perfect means bisexual. So they are hermaphrodites. That means they will have staminate and pistillate whorls in the same flower. Mind you, learners, I said staminate and pistillate whorls in the same flower, not on the same plant. Then it becomes different. If you look at the screens, you find a perfect flower of hibiscus, wherein you have a male whorl and a female whorl. But the terms dioecious and monoecious need to be understood carefully. They are the terms which are actually referring to a plant reproduction in botanical terms. Dioecious would describe a plant or a plant group where the individual plants have both male and female parts, but not both. So that means we have male plant and a female plant. If we have a male and a female plant, obviously one of them would have the staminate and the other one would be pistillate, as you are accustomed to see in papaya. We have a male plant of papaya and we have a female plant of papaya. And if you have just one female plant and there is no male plant in the vicinity, then how would the fruits be set or the seed be set? Same is the case with hemp or cannabis. You have the male and the female plants separately. And as you know from your other discussions in botany, the amount of cannabinol and cannabidiol, that means all the drugs are different in depending upon the sexes. On the other hand, the term monoecious has a description where each of the plant is going to bear both male and female flowers. Please do not confuse this with a flower having both sexes. In that case, it would be a bisexual flower or a perfect flower. Now, the plants in this case that they are monoecious, they are capable of both self-pollination and cross-pollination, and that means we would find that since the female flowers are present, you could have contrivances for self-pollination as well as for the cross-pollination. You can see on your screens the date palm, a dioecious plant which is Phoenix dactylifera. You have a male plant with a male spathe and the other plant, the female plant, which has the female flowers. Now, you could find a similar situation in case of corn, because corn is monoecious, and you would find that in a monoecious plant we have a tassel and a silk on the same plant. So, dioechy is a characteristic of a species. Either the male or the female gametes are going to be produced either directly or indirectly. We can also summarize that dioecious reproduction is basically a biparental reproduction. That means you have male plant and a female plant, and you have to pay a cost for that, because half of the population is going to directly produce offspring, because it's only the female plants which are going to produce, but then it is worth the cost in terms of evolution, because plants can only cross pollinate, the dioecious ones, and therefore there would be high genetic variation, and this would lead to an increased genetic heterozygosity, which is so very important for evolution. On your screens you can find different categories of the plants which are, say, andromonoecious, that means male and hermaphrodite flowers; gynomonoecious, which means females and hermaphrodite plants. Likewise, you have gynodioecious ones, which means female and hermaphrodite plants, and androdioecious. The basic idea is that you have different types of combinations of monoecious as well as dioecious plants. So here's a question for you: what is a trimonoecious plant? Because we have talked about the dioecious and monoecious plant. So does a trimonoecious plant exist? Well, it does. The answer would be, a trimonoecious form is one which has three distinct types of flowers on the same plant: male flowers, female flowers, as well as hermaphroditic flowers. So that means all the three variations occur on the same plant. The example is Acer, or one of the species of maple, which is Acer campestre. Now let us talk about the sex determination in plants, where we are not talking about chromosomes. We are talking about only, and there is no sex chromosome, we are talking about a single gene, and it is a single gene which is determining the sex of an individual. And there are very many examples, say Chlamydomonas, Neurospora, and even yeast, various species of yeast, that is Saccharomyces. Now in this case, since there are no sex chromosomes, we have two mating types, or two strains, as we can say. So a gene or a pair of alleles would either be plus or it would be minus. We will call this as strains. And obviously, as you must have read in bacteria and in Chlamydomonas, the sexual reproduction is going to take place when the gametes of two different strains are going to be fusing together. So the gametes of the same strain would never fuse in such organisms, and we know it's very natural that the organisms like Chlamydomonas are going to spend most of their life cycle in the haploid phase itself, and they are going to produce daughter cells asexually because the conditions are favorable. But then once the conditions get unfavorable, say for example there is depletion of nitrogen, and then now some of these cells, they are going to behave, they are going to transform their behavior, and they are going to function as gametes. The beauty of the situation is that there is no visible difference between the cells which are going to reproduce asexually and the ones which are going to behave as gametes. And moreover, the two gametes which are going to fuse with each other are also not to be distinguishable, at least by the help of a microscope. They may be biochemically different, which they are. So in this case, it is conventional to call them as isogametes because they look exactly the same. Their size, their behavior, it is the same. So they are called as isogametes. On your screens you would find that these two gametes are same as far as their appearance is concerned, but they should be classified as two different mating types. And this was established by two scientists, Ruth Sager and Sam Granick, who call these isogametes as they should be better called as mating types, MT plus and the mating type minus, so as to prove that all their behavior is the same, but then the plus would fuse only with minus and vice versa and not among themselves. A quick look on your screens would give you the life cycle pattern of Chlamydomonas, wherein we find vegetative cells which are there. Once they are going to have nitrogen depletion, they turn into isogametes, one plus and the other minus, and then they fuse, giving rise to a transitory quadriflagellate structure, and the fusion gives rise to a zygote, which under favorable conditions now germinates, and you can see on your screens it's a case of zygotic meiosis. And meiotic products are again two of the mating type plus and the remaining two of the mating type minus. So in that case we find that 4 haploid cells are going to be formed, or they are if they are motile, then they are the zoospores. So the 4 out of the 4, 2 are MT plus and the other 2 are MT minus. They may be morphologically exactly the same, but they are definitely biochemically different. How do we prove it? We prove by clumping experiments. If we add MT plus to MT plus, then there is absolutely no clumping. But if we add MT minus to MT minus again, there is no clumping. But then if you add the opposite strains, that is the MT plus added to MT minus, then there would be clumping, which means that the gametes are morphologically identical but biochemically different. So this is a very primitive type of sex determination. On your screens you can find a pictorial depiction of the plus not clumping with the other plus and mating only with the opposite strain. Likewise, the minus also does not clump amongst itself, but clumps or pairs with the opposite strain. So it is some sort of a invisible primitive sexual differentiation, and there are no visible differences in the morphology of the gametes. Now recent studies have shown that Chlamydomonas reinhardtii is actually a haploid, and it has a nuclear genome which comprises about 17 chromosomes, and the total size is 110 megabase, and it has a 203 kilobase chloroplast genome and a 16 kilobase mitochondrial genome with 14,000 protein coded genes. Let me remind you learners that the megabase here is equivalent to a million nitrogenous bases, which is roughly equivalent to 1 centimorgan. So the recent studies have also shown that what we were talking about, the MT plus and minus, they have been found, this locus has been found on chromosome number 6, and the gene has been identified that mediates the expression of MT minus mating type, which is essential for the cell fusion is in response to nitrogen depletion as to when the cell would transform into a zoospore. So just to summarize, we find that the Chlamydomonas gives us a very good system of sex determination, which is not an apparent one. It is a hidden one, but nevertheless it is there. In our next lecture, we would be extending this whole discussion and going over to more examples of the monogenic sex determination, and then we would go over to the chromosomal basis for sex determination in plants. Thank you.